Okay, so let's start. We're at the top of the hour. Today we are speaking with Mai Tran. She is in the Ross-John Laboratory as a postdoc, and her title of her talk is Mechanism for Cross-Reactivity of T-Cell Receptors with Insulin C-Peptide and Hybrid Insulin Peptides, HIPs, presented by HLA-DQ8. A little bit about her. She commenced her PhD in Professor Jamie Ross-John's laboratory in 2018 at Monash University in Australia. Her research was focused on understanding the structural biology and protein biochemistry that leads to autoimmune disease and type 1 diabetes. After completing her PhD in 2022, she continued as a postdoctoral researcher in the Ross-John Laboratory, where she investigated the molecular basis of human CD4 positive T cells, cross-reactivity between self-antigen and altered self-antigens, peptide fusion by post-translational modification implicated in type 1 diabetes. I will drop her recent paper in the chat for people's perusal. And thank you so much, Mai Tran, for joining us. Look forward to hearing more about your work.
Okay, thanks, Monica. So today I'm going to present my project, which is I completed in 2 years as a postdoc. So let's move forward. So type 1 diabetes is characterized by immune system target again the insulin produced in beta cell in pancreas. So that leads deficiency of the insulin produced by the beta cell. And when the individual diagnosed with the T1D, or mostly patients have like 80 to 90% beta cell mass were destroyed. The gene that involved is MSC class 2 gene called DRDQ are strongly linked to develop a T1D, especially in individuals with a haploid type like DR4, DR8, DR3, DR2 have a much higher risk to develop a T1D. However, most T1D patients have DQ8 or DQ8, DQ2 haploid type expression. Whereas protection molecule like DQ6 or haploid type DR15, DQ6 are protective molecules. When the individual diagnosed with the T1 clinical onset is there multiple or antibody of the isolate autoantigen detected before clinical onset, like insulin, GAPs, gene transporter 8. So this suggesting the beta cell release on antigen. So this antigen uptake by C4 T cell and present for MC class 2 and trigger immune response target a beta cell that lead beta cell death. So is MC class 2 a receptor is bound to the peptide and displaced on antigen present cell surface for C4 T cell recognition. And C4 T cell recognize peptide MSC by 6 loops. 4 of them are germline encoded, like CR1 and 2. CR3 alpha and beta look very high diverse. So MSC class 2 recognize peptide at the position P1, P4, P6, and P9, especially DQ8 is more susceptibility for type 1 diabetes is because of P9 by the pocket, which is refer acidic residue. They can form a salt with arginine. Whereas the protective molecule like DQ6 is this binding pocket of because aspartic acids on the DQ8 alpha change. So they only accommodate smaller residue on the P9. So is every mature insulin produced or also associated with release C-peptide. So this C-peptide was found fused with highly expressed protein in granule and generate non-antigen we call it hybrid insulin peptide. So this hybrid insulin peptide is shown strongly respond by human C4 T cell isolate from isolate of origin of donor organs who had T1D. And you can see that these 2 clones here show strongly respond to HIP peptide, which is C peptides fused with IPP2, and weakly respond to C peptide. Because C peptide, you can see they lack anchor residue at position P9, whereas HIP contain glutamic acid at P9. So how the DQ8 present HIP recognized by C4 T cell is poorly understood. So in the 2015, we saw been published in in diabetes journal. She show is this C4 T cell clone isolate from donor patient donor organs, So respond to the C-peptide, and the gene usage is dominant at TRIV26-1 and TRBV5-1. So this is very similar observed. We studied, we were published in Nature in 2021. We used DQ8 HIP peptide and we isolate C4 T cell from PBMC of 3 patients just recent onset T1D. And we also observe a similar gene usage at like TIV26-1 and TRBV5-1, especially the second patient. So highly frequency of TRIV26-1. And interestingly, we found that the patient number 2 have a clone almost identical with the C-peptide responsive C4 T cells, and only different amino acids on CR1 amino acids on CR3 beta loops. And another clone, it's ET650.4, also have similar motif compared with 2.13, was published in 2015 in diabetes. So we able to understand the cross-reactivity with the HIP peptide. So we perform a stimulation assays with power BLCLs, which express as IPC, express DQ8 on the surface. And we power with the peptide HIP or C-peptide and overnight. And after that, we culture transduce TCR, which express TCR on the surface or SKW3 TCR line. And we, after 20 hours, we stand with the CD3 and CD69. And we found this, the C4 T cell, which is C-responsive, T cell for T cell in the top panel, and HIP responsive T4 T cell in the bottom panel. They're very sensitive to the HIP peptide, as you can see, compared with a C-peptide, which we published in 2021 in Nature. So able to do this C4 T cell can cross-react to multiple HIPs. So we select like 3 HIPs, which is C-peptide fused with IPP2, we call it HIP1, and IPP1, we call it HIP2, and neuron peptide wipe, it we call HIP3. We select 4 C-peptide responsive C4 T cell, we published in 2015 in diabetes, and we also select 3 HIP responsive C4 T cell clone. And we performed stimulation assays similar I described previously. And we found is 7 out of 8 selected C4 T cell clones so respond to multiple HIP. You can see it is no peptide in the first column and the C-peptide in the second one. And HIP1, HIP2, and HIP3 is from the third to the fifth. As you can see, some of them can rush like ET650.4 can rush react to multiple HIP like 1 to 3. And similar to that observed in ET650.5. And we also confirm this TCR so strongly respond to C-peptide converts to mutate for position P9 to glutamic acid. So suggesting this is the HIP peptide is more potential antigen for DQ8. So we also perform.
May, could I just qualify something on that slide? On the previous slide? Are you saying this is a better binder? The G90 is a better binder than insulin C8?
No, because C-peptide lacking acidic residue at position P9, and we mutate to glutamic acid. You can see the cell respond much better. So to confirm,
yes, better. Okay, great.
It contains glutamic acid P9 more potential than C-peptide.
Great, thank you.
And we perform the SPR, and we found similar like stimulation assays. They have the TCR, so multiple respond to multiple HIP. And in this tremendous talk, I focus on the 2 TCR, 2.13 and ET650.4. Now, these 2 TCR share the same gene, TIV26-1 and TRBV5-1. And the difference of these two TCR is TCR3 alpha and TCR3 beta, different sequence. And TCR2 beta loop have similar motif on ET65.4, except in the residue like arginine in 109, glutamic acid, and long one residue compared with 2-13. So I saw the structure of 2-13 in complex with DQI at the bottom and peptide in the middle and TCR in the top, in complex with C-peptide on the left, HIP1 in the middle, and HIP2 on the right. So this is TCR footprint to show is the TCR ROS, this 2-13 ROS reactive C-peptide and HIP is biased TRAV20 and TRBV5, that's one. This is similar bias observed in TCR 1.9 and 2.13. They were published by us in 2015. And the same TCR it was published by Yan Wan in 2019 in Science Immune. So this is biased by beta framework, CR1 beta and CR2 beta loops. And this is superimposed of TCR with C-peptide, HIP1 and HIP3. As you can see, the identical conformation of residue on CDR3 loop or CDR loop general. The interaction also very similar. The difference I observed here is when the 2-T interact with C-peptide, the arginine or CF1 beta loop have upward conformation. When they rush to HIP1, it's almost skewed toward the peptide. So they fall interact with the PI valine. And because the valine is no longer residue at the PI, so CR2 beta loop is still away from peptide able to fall interaction with the PI valine. Whereas the alanine in C-peptide is shortest residue, so it seems like it moved forward. When it's recognized with the HIP3, the arginine 37 or CR1 beta loop is skewed more closer to the peptide and form intensely with the peptide and the GQI. So I also show structure of the ET65-4 ROS react in complex with the HIP1 and HIP2. As you can see, the footprint of the TCR on the QI is almost highly conserved patterns. And we found this TCR not by a TRBV5, that's one, but by a TRIV26, that's one, which is observed on the TCR on celiac disease. I'll share the same gene TRIV26, that's one, which alpha framework, CR1 and CR3 beta loop interact with arginine 73 in DQI beta chains. So this is superimposed of two structure of ET65-4. You can see it's almost identical interaction, and the conformation is different on arginine 109 because it's on the HIP2, the PI residue is isoleucine. So they change the conformation able to form interaction with residue on the PI. So I compare with these two TCR to both 6504 and 2.13 in DQI HIP1. And the footprint is highly distinct compared with two structure. And this is the TCR interact with the peptide, as you can see, ET65-4 on the top and 2.13 in the bottom. The CR3 alpha loop of ET65-4 form intensely interaction with the peptide from position P-2 and from P2. And they always interact with position from P7 and to PI and to the main chain of ranking residue. When you look at 2.13, they less form interaction with residue on N-terminus compared with ET65-4 because CR3 beta loop is shorter than ET65-4. The arginine is different. One is, I'm sorry, this the arginine, sorry, what glutamic acid 109 sit on the top of the peptide, whereas the arginine 109 of T6 is still away. So this is another movie I can show you how the T6 interact with the HIP1. You can see the PI residue is sit underneath of arginine 109 and arginine 37. So there's more space on this portion PI. So this T6 can accommodate longer residue like valine, isoleucine. Whereas it is 2.13, the glutamic acid 109 is sit above of the peptide, and PI valine surrounded by CR1 and CR2 and CR3 beta loop residue. So they only accommodate the smaller residue at the PI's position on the peptide. So I perform alanine scanning on the peptide. As you can see on this one is 2.13 in the C-peptide at the top and HIP1 in the middle and HIP3 at the bottom. As you can see, when you mutate at P2 and P on the C or HIP1 3 is the impact on T cell response. You can see the interact intensely at P2 losing because P3 is they may interfere. They don't interact with the TCR, but when you mutate P3, they may interfere with phenylalanine 58, may interrupt with the CR3 beta loop. There's no impact on to the HIP3 from position P6 to P12 because that is where it's the joint between the HIP peptide, the difference of P6 to P12. In the C-peptide, only we observe it impact the T cell response, its position P6 from proline to alanine. But interesting in HIP1, when we mutate to valine to alanine PI, the enhanced the 2.13 T cell response. As I can sort of, as I saw you structure before, the CR2 beta loop move away because the valine longer side change. When it becomes a smaller residue, this phenylalanine move closer, they gain more interaction with the QI alpha change. When it moves away, they lost interact with the QI alpha change. So that can explain why they can enhance the response when the valine mutate to alanine. And for the ET6 it's when you can see that CR3 alpha loop form intensively on N-terminal from P-2 to P2. When you mutate from P-2 to P2 to alanine, it's a significant impact on T cell response on InsC-peptide, HIP1 and HIP3. Interestingly, we found is when in HIP3, when we substitute position P5 to alanine or a certain to alanine at position 7, it's enhanced T cell response. Because the arginine 109 is still away from the peptide, it's more close to the QI beta chain is where the the position of residue like P6. There's more potential impact on the arginine 109 interaction. So we look at another HIP is C-peptide filled with chromoganin 8, which contain smaller residue at P7 and longer side chain at P8. So we want to test hypothesis. Our hypothesis is with the smaller residue at P7, as we see on in alanine scanning, enhanced T cell response, and can accommodate longer residue at P8, which is glutamide. So we perform stimulation assay as we predicted 2.8 to 2.13. This is not cross-reactivity HIP4, which is the peptide response with chromoganin 8, whereas ET6 can cross-react to HIP1, 2, 3, and 4, which is contain longer residue at P8 and small residue at P7. So this is summary. We found is 5 out of select 7 in transient SKD3 T cells so respond to multiple hits. And 213 show the bias, TRBV5-1, and why ETC5-4 bias, TRAV26-1 gene usage. And we saw the CR3 loops are the key factor to determine the level of T-cell cross-reactivity. As you can see, the different level of cross-react between 213 and ET65-4, only different in CR3 loop. And we found the germline encoded arginine 37 and OCR1 beta loop and non-germline encoded arginine 109. OCR3 beta loop play important role in cross-reactivity. So I would like to thank my co-supervisor, my supervisor, Timmy Ross-John, and my group leader, Hugh Reed, to allow me to involve this project and guidance and support. And thanks for all Ross-John lab members. Also thanks for Stu Mandarins to provide the T-cell load. And thanks to you to have me here for my presentation. Thank you.
Thank you very much, May. That was fascinating. I wanted to see if I'm going to open it up for questions to people. Feel free to unmute yourself or raise your hand if you need help, or you can drop something in the chat.
Hi, May, Stuart here. Thank you. That's a beautiful presentation.
Thank you.
Can I ask, have you measured the HLA binding affinity independently of these peptides? Sorry.
No, because we press incorporated link.
Oh, okay. Yeah, okay. So
And
you haven't
started DQI resistant with DM.
Yeah. So you haven't. I was just curious if there's a difference in binding affinity of the different HIPs to DQ8, but you haven't addressed that. Okay, thanks.
Yes.
Thank you for that question.
So, actually, I have a small question. Maybe it's quite out of my knowledge. So, based on the when you activate those specific T cell clone, I just want to know the phenotype. So, just because we know for some of the conventional antigen, which can increase the tolerance immunity, I just want to know that those T cell clone become the cytotoxic T cells or still have some of the features of the tolerance T cells phenotype?
That's a great question.
Yeah, and thanks for the question. For the T cell, for T cell, we isolate from the patient. I don't have information for the phenotype of this clone, but for the C-peptide responses for T cell were published in 2015. Most of them is T helper, T4 T cell, as I in memory so far. Yeah.
Okay.
Can I just chip in there, Mike? Yeah, to the best of our knowledge, they're not cytotoxic. We haven't tested them directly for cytotoxicity, but the CD4 positive memory T cells, as far as we know.
Okay. Thank you.
Thank you. I had a quick question regarding the sort of the last one of the last slides you showed, May. Your summary slide. Can you go back to that? Yeah. So your germline coded arginine 37B and the non-germline coded arginine 10-9B play important role in cross-reactivity. How would you weight those, the non-germline coded versus germline coded, in this particular sort of experiment, and like in the larger view, if you've seen more data that suggests, do you feel like there's a real germline footprint that is supersedes or is greater than the non-germline footprint or not?
Yeah, for in the germline encoded arginine or the one beta loop, we observe in this TCR, you can see they have different conformation when they cross-react to HIP. And for the non-germline 109, we not only observe in the TCR, we also observe the TCR on celiac disease as well, especially position 109 on CR3 beta loop. They play important role. for cross-react and cause the disease were observed. Yeah.
So can you expand, like, can you expand to your wider data, I guess, set, or are you able to in terms of germline versus non-germline, which seems to have a greater impact? Or can you not
Yeah.
say that?
Yeah, that's interesting. We would like to see more on the T cell, particularly in Taiwan diabetes. We want to know any similar motive, especially I think in 109, CD3 look on different patients or different TCR.
Right. You
Yeah.
have to get more patients. Yeah. Yeah, it's a very interesting finding, and it really kind of gets to the heart of germline, non-germline. So at least in this particular data set.
Yeah.
Okay. I noticed a couple of genetics scientists or base scientists who run big sort of labs at UCSF and also at Indiana were on the call earlier, and perhaps I'll reach out to you separately for conversations about this interesting new result that you've brought to bear. And we really appreciate you taking the time coming to us from Monash University in Australia to discuss this really interesting work. Thank you so much, May. And we'll look forward to speaking again in the future.
Thanks for having me, Monica.